Yes, all, and very warm welcome to the session. Yes, okay, everyone, fantastic. So today we are going to make a detailed look into this DBT set of questions, that too from our RDNA technology. So this is myself, Dr. Saranya. I am speaking to you about the most important DBT questions, that too it is from the RDNA technology paper. Right, so these are the previous learners who have already cracked up their examination. So the list is pretty high. Even this time we have got a very good result. I seriously want everybody who is watching this lesson to crack, right? Okay. So these are the current courses they have that I have launched in this Plus Unacademy Plus platform. Okay, so I am doing a jumbo DBT practice course. So that is really a very important course for you all. Okay, so likewise I am doing one more important molecular biology master course. So this is also a bit important course for your preparation. Yes, so good afternoon everybody in the session who is here. So let us make a very important thing. Right. Okay. Fine. If at all you are new to Unacademy, you can use my code Saranya Live, such that you will get a 10% off on the total fees. Fantastic. Come on everybody. So let's begin the first question of the day. A novel type 2 restriction enzyme. Okay. So there is going to be a novel type 2 restriction enzyme. Yes, Anthony, Ram Prabha. Yes, Arya. Okay. Right, so this has been isolated, right, from the thermophilic bacteria. Okay, right. So this restriction endonuclease, it is going to recognize the particular sequence. They have given the sequence 5 prime A T A A N N N T T A T C 5 prime to 3 prime. So N means any nucleotide. And what this does, it is going to cut after the third A. Okay, so exactly after this third A, it is it is going to make a cut. So remember, this is the first A, this is the second A, this is going to be the third A. Okay, right. So here it is going to make a cut in the above sequence. So what is the fate of DNA after restriction digestion? Okay, so what things it is going to contain? So that is the question. So shall we all try it out to find the answer for this question? Yes, everybody think and answer this question. Yes? Yes, Anthony. Fantastic. What about others? So what is going to be the fate? So see here, 5 prime. Yes, Anthony, correct. Very good, Ram Prabha. 5 prime A T A A N N N T T A T. This is 3 prime. So obviously there is going to be a one more strand. Yes, Deepika. So this is going to be T A T T. Yes. This is going to be AATA. Okay, so this is going to make a cut here, right? Fine. So what is going to be this 5 prime overhang? How many nucleotides long? Yes, it is going to be how much long? 1, 2, 3, 4, 5, 6, 7. So it is going to be a 7 nucleotide long 5 prime overhang. Got it, everybody? Is it now clear to you of how this is obtained? Yes? Okay, fantastic. So this is what is called as an overhang. Okay, right, absolutely. So the correct answer is going to be your answer B. Fantastic. Pyro sequencing is a very important and interesting method. Okay, pyro sequencing. Sorry. So this pyro sequencing, it is going to derive its name from the fact that, okay, so what is the fact that is telling this is going to be the pyro sequencing? Okay, so it says that, yes, it says that the bases, it is detected by means of pyrolysis it uses the enzyme A pyrase to detect the bases. It detects pyrophosphate released during the base incorporation. It generates pyrograms as output. Which could be the correct answer? Yes, everybody in the class. Think. Why it is called as Pyro Sequencing? Correct. Pyro Sequencing is a sequencing method, right? And what it detects? It detects the pyrophosphate. Correct? Absolutely. No other doubt. Yes. So definitely answer C is going to be the correct answer. Yes, Mukesh, Deepika, Ram, Sayantani, absolutely correct. So pyro sequencing means you could have remembered the order, isn't it? So first you have your DNA N, right? Along with that you are going to add your DNTP. Okay, so it is going to extend your DNA plus 1. Along with that it is going to release a PPI. Yes, Snehal, correct. So this PPI, it is going to react with your luciferin. Yes, it is going to undergo so many actions, and finally it is going to produce your ATP. This ATP will react with your luciferin and it will form the light. Yes, Mukesh, very good afternoon. Hence this is going to be the correct answer. So pyrolysis is a different process. Okay, it is not at all related to us. It uses the A pyrase to not to detect, it is to degrade the base, isn't it? So unwanted fellows, it will be degraded. So A pyrase is used, but the use is not to detect the base, but it is useful to delete the unwanted stuff. Okay, so it detects the pyrophosphate. Absolutely, this is the most suitable answer for this question. Yes, it generates the pyrogram as output, not at all. Okay, right, good, fine. So active site of the serine protease, it is going to contain which of the following, right? Yes, everybody, come on. Yes, ready or what? Done. Active site of, very good. Yes, Vishu. Sayantani, Deepika, exactly correct. So your serine proteases, it is going to contain your serine residue, histidine residue and aspartic acid. Okay, right? So aspartic acid, serine, histidine, aspartic acid. Exactly, this is the active site of all the serine proteases. Okay, so what is the active site of serine proteases? Serine, histidine as well as aspartic acid. A very, very important and a direct question. See, one more similar question. Which one of the following is present in collagen? Which one of the following, it is what is present in the collagen? Give a try. Very good. Fantastic, everyone. Keep it up. So, absolutely. Yes, Aishwarya, Vishu, Ramprabha. Correct. It is going to be glycine followed by X and Y. Yes, Siji. So, X means it can be like your hydroxyl proline. It can be your hydroxyl lysine. Okay, exactly. So, that is what is going to be the correct answer for this question. Okay, so let's make a move towards the next question. The electrophoresis of a purified protein, okay, in the SDS along with the presence of 2-mercaptoethanol, it is going to yield 2 bands. Okay, one it is going to be 35 kilodalton, other one is 45 kilodalton. Okay, one is 35, other is 45. However, in a gel filtration chromatography, the same protein, it elutes as 80 kilodalton. Okay, so this is in SDS, this is in gel filtration chromatography. Okay, so in gel filtration chromatography, it is eluted as 80 kilodalton. Okay, right. So what is the conclusion that you can draw from this experiment? Okay, the protein, Sanarnya. It is not purified to homogeneity. The two bands generated in SDS, it is due to degradation. The protein is a multimer. It is a heterodimer. Very good. Come on, everybody, give your try. Sanarnya, Vishu, Ram Prabha, they all gave the try. Good. What about others? Come on. Did we right or wrong? Give a try. Yes, Siji. Good. Fantastic. Yes, Deepika. Awesome. Correct. So, this is going to be absolutely a heterodimer. What do you mean by dimer? Something is made up of two subunits. Yes, Aishwarya, I'll tell you. Yes, Vandana. Good. Yes, Mukesh. Very good. If it is made up of two subunits, it is what is going to be called as a dimer. When it is going to be two subunits? Yes, because when you perform them the SDS page along with Chandini, when you perform them the SDS page in the presence of two mercaptoethanol. So this mercaptoethanol, what it will do? It is going to reduce your thing. Okay, reduce means it is going to cut off all your bridges, isn't it? So actually it could have been a dimer. This bridge is released, then it is showing a single subunit. Correct, Prachi? So it is going to give you the single subunit. So that is why you got two subunits. If the subunits are having the same molecular weight, for example, original fellow is 80 kilodalton, this is 40 and 40, then you may think it is a homodimer. Okay, right. So if it is going to be 80, I got fragments like 30, 25, 25. Yes? I think 2 or 3, Prachi. So if I am getting like 30, 25, 25, absolutely 3 units are there. So it is a trimer. Okay. So out of this 3, this fellow is quite different. So you cannot call it as a homo. So now also you call it as a heterotrimer. Okay. So one more example I will give you. So like I have 100 Dalton. I have got 25, 25, 25, 25. This is what my output when I do this mercaptoethanol. Okay, right. So this is going to be a tetramer. Okay, so this is going to be a heterotetramer. This is going to be a homotetramer. This is going to be a trimer. This is going to be a monomer. So 4 are there. So it is a tetramer. Correct. How are the 4? All the 4 are equal. If all the 4 are equal, then you have to call them as homo. So it is going to be a homotetramer. Understood the logic how to solve this question? But in this question, it is not same. Okay, so one is 35 and other is going to be 45. Okay, so 2 sub units. So it is going to be a dimer. Yes, Lipika, got it now? Vishu, very good. So it is going to be 2. So if it is going to be 2, it is going to be a dimer. Yes, if it is similar, it is a homodimer. If it is going to be different, it is going to be a heterodimer. So in this question, it is different. Yes, Prachi. No, it is going to be a heterodimer. Understood? Okay. So this mouse model, yes, Aishwarya. Good. Yes, Lipika. The mouse model for type 2 diabetics. Okay, so the mouse model that you use for type 2 diabetics, it is NEZZ mouse, skid mouse, nude mouse, or nod mouse, which is used as a diabetic model. Okay, right. So there are variety of mouse models you commonly use in your labs, right? So which is going to be for your diabetic? Come on, everybody in the class. Yes. What is this nod? It is non-obese diabetic mice. Okay, right. Absolutely. So that is going to be the correct answer for this question also. Got it, everybody? Yes? Okay. So what is this skid mice famous for? I told you NOD is non-obvious diabetic mice, right? And alternatively you have this skid mice, isn't it? Many a times you could have heard about the skid mice. Skid means it is going to be the severe combined immuno, all the names if you know, major problem is done, deficient mice. If you know the acronym, then it is halfway done. Yes, so this is especially for you take the human cells and you develop this lymphoid organs and then you put them inside this immunodeficient mice, right? So that is what it is, all for this immunodeficiency. Okay, right. So that is going to be the skid mice. Okay, and then what about this NZB mice? Come on, everybody. NZB is nothing but New Zealand, isn't it? New Zealand black mouse. Okay, right. So NZB is nothing but it is the New Zealand black mice. So it will be combined with some other strain also. Yes, then you can use it in variety of things. Okay, so if it is going to be combined with this NZB, if it is combined with WF1 bar J, then it will become an autoimmune. Okay, so like that they have a variety of strains. Okay, so this is the basic thing. Okay, basic model. And what about your nude mice? Come on, everybody. So what about this nude mice? So this is also an important mice. Yes, so because this nude mice, it has a mutation. Okay, so because of this mutation, the thymus, it is going to get absent. Okay, right. So because and also they have no hair. Okay, so that is why it is called as nude. Right, it has no hair. Okay, right. Yeah, so they lack a thymus and they have no hair. So that is the significance of this nude mice. So whenever you are reading this, all the relevant things. Yes, absolutely. ADA is going to be your adenosine deficiency mice. Correct. So it is not thymectomice. It is all your strain based one. Okay, so you are going to create such things inside. Understood? Yes, everybody. I hope that this is all fine. So we will make a move towards the next one. But diabetes, it is going to be NOD only. Right. So the next question. Yes. So a hypothetical relaxed circular plasmid. Okay, a hypothetical relaxed circular plasmid. Okay, so it is present and this is going to be 4500 base pair. Okay, so relaxed circular plasmid. Okay, so relaxed circular plasmid, right? So this is going to be 4500 base pair in length, right? If for supercoiled form, the twist, okay, so the twist is going to be 440, okay, and the width, it is going to be -20. So what is the plasmid considered to be? So what the plasmid is considered to be? So you have to divide and find out what is the for your bDNA. Yes, absolutely. Think for your bDNA, what is going to be your actual number per turn? Yes, yes, Anthony. So for bDNA, what is going to be the number of turns that is present? Quick. For bDNA, how many base pair? 10.5. Okay. So actually speaking, you have to divide this 4500 divided by 10.5. Okay, right. So you should have got how much? 428 points. Okay, so this is what the number of twist you should have got. Correct? Yes? Absolutely. So you are going to divide, you will get the number of twists. So actually it should have been 428. Okay, so now what happens? this twist is going to be 440. Okay, right, and the width is going to be minus 20. So total, what is the linking number? It is going to be 420, isn't it? Now this linking number is going to be twist plus width. So this is 420. So this is greater than my expectation or below my expectation? Think of. Yes? Yes, absolutely it is going to be below my expectation. So if it is going to be below, absolutely it should be your negatively supercoiled. It has reduced. Instead of being 428, it is only 420. That means it has reduced. So if it is going to be reduced, that means that it is negatively supercoiled. Got the logic? Everybody? So the key point is you have to know that for BDNA it is 10.5. Okay, right, 10.5 base pair per 1 turn. So how many turns it is there? Hypothetically, it should be 4500 divided by 10.5. Right, yes, so that is coming around 440. And here they have, that is coming around your, this thing. Okay, so that is coming around this 428. Yes. Oh, sure. Okay, I'll take it, Prachi, don't worry. The next session I will make it, don't worry. Alright, so this is clear? Okay. So this restriction enzymes that is produced by E. coli, they do not cut the self DNA. Why? Immunity to the self. It is going to be none other than your DAM methylase. Okay, so DAM is nothing but it is going to be your DAM methylase. Okay, so it is clear now? Alright, good everyone. So the next question, it is regarding the enzyme that is used in solid sequencing technology. What is this? Under NGS, you have a variety of techniques, right? So under the NGS, you will be having many more techniques. Okay, right? And number one, it is going to be your 454 Pyro Sequencing. Okay, right? Number two, it is going to be this solid method. And then it is going to be this Illumina method. Okay, so these three are the key methods that are usually used. And every year, one or the other question will be coming. Okay, right, fine. So in this question, they are asking the enzyme that is used in this solid sequencing. It is going to be what? Sequenase, polymerase, ligase, tadpole. Fantastic. Yes. So this Illumina is sequencing by synthesis. This solid, it is sequencing by ligation. Absolutely. So ligation means it uses ligase. Polymerase is usually PCR related. Okay, yes, and sequenase, we are not using up here. So the enzyme that is used in solid sequencing, it is going to be the sequencing by ligation, and absolutely it is none other than your ligase. Okay, so coming to the next question, this E. coli, it is having a mutation. Okay, right. So this E. coli, it is going to have a mutation. Okay, right. And this mutation is taking place in the operator region. Okay, of this lac operon. It is containing the suppressors. Okay, right. Okay, so it is containing the mutation in this operator region. Okay, right. So what will happen? Is it going to produce your beta galactosidase? Sure, Prachi, I will. So it is going to produce the beta galactosidase even when the lactose is absent. It is going to produce beta galactosidase only in the presence of lactose. Yes, Tanmay, it will not produce beta galactosidase. It will produce even in the presence of glucose. Which is going to be the correct answer for this question. Come on, everybody. Yes. Absolutely. A is going to be the yes, Siji, correct. Yeah. So absolutely. So this is going to be your yeah. So this is going to be your it is continuously produced even if your lactose is absent. Okay, right. So this is under your inducible operon. Okay, but this is only going to be under your constitutive operon. So if your operator is mutated, your repressor can never come and bind. Okay, yes, absolutely, yes. So that is why it is always on. And why I didn't choose this? This is something regarding to the cyclic AMP. Okay, so if your glucose is present, okay, so if the glucose is high, cyclic AMP will be low. So when cyclic AMP is low, then your cyclic AMP activating protein, it will be low. So your RNA polymerase will slip. Okay, so this will not allow your your galactosidase production. But this is the apt answer for this question. Okay, so I think Prachi has got a doubt between what is an ortholog and paralog. Okay, so see here, paralog. Paralog means, yes, you have to remember the clue PSO. Ortholog clue is ODO. Okay, yes, okay. So what is this paralog? Paralog means it is going to be the same organism and it is going to have a different gene. How this could have happened? Maybe there is the event of sure, I will take Prachi, don't worry. Numericals of enzymes definitely we will do. Yes, so this PSO, no, it is the paralog means it has undergone some kind of gene duplication. Okay, yes. So this only makes them to act as a paralog. So what could be the example? You take the human. Okay, yes, Uzair. In human, you have your delta hemoglobin, gamma hemoglobin, epsilon hemoglobin. Yes, so these are all the different, same gene. Okay, but it has undergone the small duplication. So the gene, it has become little bit different. Okay, yes. So the gene, the same organism, you have the different gene. This is what is called as a paralog. Ortholog means the organism, it is going to be different. The gene, it is going to be the same. For example, okay, so for example, if you take this insulin gene, so insulin gene, it is present in cow, it is present in human, understood? So this is what is going to be the ortholog. Now you got it, Prachi? Yes? Okay, fantastic. So RT-PCR reaction, it uses which of the following? RNA dependent DNA polymerase and DNA dependent DNA polymerase, RNA dependent DNA polymerase and DNA pol 1. Yes? Yes, absolutely. Your RNA polymerase and DNA dependent DNA polymerase, RNA polymerase and DNA pol 1. Exactly. Come on, everybody. So what is the sequence of usage of my RT-PCR? So in RT-PCR, first what you use and then what you use. Fantastic, everybody. Absolutely correct. Yes. Very good all. Very good. Exactly. Yes. So in RT-PCR, you have an RNA dependent. RNA dependent means RNA is used as a template. Okay. So it is an RNA template dependent DNA polymerase. Okay. So like our telomerase, isn't it? Yes. So like that this fellow is doing. Okay. So it is an RNA dependent DNA polymerase. And the other one is going to be the DNA dependent DNA polymerase. So this is the next thing. So first it does this reverse transcription, then it is going to amplify your cDNA. So this is going to be a DNA dependent DNA polymerase. Okay, and always never confuse your DNA polymerase, they can make use of your RNA primer. Okay, yes, it makes use of RNA primer, but always remember DNA polymerase is using the DNA as a template. Okay, so template name only should come here. So DNA polymerase, they will be always DNA dependent. Okay, your normal polymerases. The normal polymerases that we use for replication, it is going to be DNA dependent DNA polymerases. Got the point, everybody? Okay. The linear and circular forms of your DNA can be distinguished using which of the following? Come on, everybody in the class, give a try for this question. Yes, so sir, everybody think of an answer. Very good. Yes, Prachi. Got it all? I have a linear form, I have a circular form. I want to distinguish them. Good try, everybody. Yes, Anthony, good. Yes, Azay. Fantastic. Think, everyone. So here, you can get an explanation for everything. Yes, Lipika, correct. I'll tell you why it is going to be this D. Okay, right. So see, if at all I am going to have my linear DNA. Okay, right. So, and there is going to be my circular DNA. Yes. Okay, right. So I am having a linear DNA and I am having my circular DNA. Okay. So if I cut by my exonuclease, my circular DNA, can it be cut? Any possibility? Exonuclease means it will cut only at the end. Correct. This circular DNA, can it be cut by your exonuclease? Will I get any band if I cut them by exonuclease? Any possibility it is there? Yeah. Yes, you should thank DBT only. They can never cut, no? Isn't it? Exonuclease means they need the end. Right? So your circular forms cannot be treated by exonuclease. Yes, Jen. So only your linear form can be done. What about this 260? 260, and you are generally, as far as your absorbance is considered, it can distinguish between your single strand and double strand. Okay, but you can understand that even though if your plasmid is circular, yes, it is double strand. If it is linear also, it is going to be double strand. Correct. So maybe this cannot make a major distinction between them. Got it? Yes. But if at all you are using your gel, okay, in gel electrophoresis you can separate them, because you know that linear will be at the top, your plasmid will go to the bottom. So in a gel you can do, okay, but here you cannot do. Okay, so why I couldn't choose my endonuclease? See here, endo means it is inside, correct. Here also I have got an inside, here also I have got an inside, no? Understood? Yes or no? So the site, okay, so the sites may vary, correct. For example, two sites are present here and here. Okay, one site is here, one site is here. Okay, so here I have the same two sites. Okay, right. So anyhow it is going to digest, at the 2 points. Alright. So it may not able to distinguish correctly. Maybe it is forming, like for example, if I cut it, this is 10, this is 10, this is 10, this is 10. Okay, so it will all be a monomer. So you will get at a single band at 10 kV. Okay, so here you will get 3 band. Yes. Okay. 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 Right. So we'll go to the next question. Good. Right. So this protein-protein interaction, yes. So this is evaluated by all of the following except which of the following ones? Come on, everybody in the class. Now it is visible. Fantastic. That's it. Yes. So chromatin, it is meant for your DNA protein interaction. Western blotting is between your antigen and antibody. Okay. So that is going to be your protein-protein interaction. Okay. And yeast to hybrid, this is also protein-protein interaction. Co-immunoprecipitation is also protein-protein interaction. But this fellow is only the except fellow. Okay. Right. So if you were to use this E. coli DNA polymerase instead of your tag polymerase, you have to, what you should do? Come on, everybody. You have to use your E. coli DNA polymerase, okay, instead of your tag polymerase in your normal PCR reaction. So what you should do? You should add a fresh enzyme after each denaturation step. You have to carry over the denaturation step at 50 degree instead of 95. You have to use the different primers. You have to use a water bath instead of thermal block. Good try, everybody. Come on, come on. Fantastic. Yes. Got it. Good. Only logical answer is A, isn't it? Can you carry over denaturation at 50? 50 means it is annealing, isn't it? It is not for denaturation. Gone. Right. Different primers. No. Only this is going to be the relevant answer. Got it, everybody? Correct, no? Very good. Fantastic, everyone. Keep it up. Keep it up. Good. So, in PET vector, PET is a good expression vector actually. Okay. So, it is going to have a high level of expression of the cloned gene. So, which of the following promoters you will be using up? Okay. So, you are going to use which of the following promoters? Alright. Come on, guys. Don't worry. So, this is going to, yes, Lipika, good, don't worry. One or few questions will be like that. It is going to use your T7 promoter. Yes, Nehal, good. PET vectors, they use T7. T7 is bacteriophage promoter. It is very, very strong promoter, so they give the high level of expression. Okay, so even that can be controlled by temperature. Okay, right. So, which one of the following technique can be used to determine alpha amylase gene polymorphism? Very simple question this is. No? Fantastic. Everyone. See, gene. Gene means which you have to use, which blot you will use for a gene. None other than your southern blot. Yeah, absolutely. Gene means DNA. DNA means southern. What is this dot blot? Dot blot means it is a crude method where you take the total stuff. Okay, so you are not going to use specific SD. Usually the routine procedure is you have to take the crude protein. Okay, yes, you will perform your SDS page. Okay, and then you will take one specific band. That band you will be transferring into this blotting paper, and then you will be adding the antibodies. This is the usual procedure. Okay, in dot blot you don't have a time. Okay, so what you do, you take the total crude protein as such, and then you do this antibody addition. Okay, So whether it will give a rough idea whether your protein is present in the sample or not. If protein is present, you will see a band. If not, you will not see. So this usually used for screening. Okay, so you have 200 samples. Among the 200 samples, you want to know which is going to contain your sample. Okay, for that crude method, you can use it for screening. But you want to confirm this thing and all, you have to go for the Western blot. So that is the dot blot. Northern, it is meant for your RNA. Slot blot is different. Southern blot is only for the gene. Okay, so hence A is the answer. Good. Yes, Mohammed and Sheenal, Siji, Lipika, Prachi. Very good. So which one of the following transgenic plant occupies the largest area in the world? Come on, everybody. Give a try for this question. Largest area in the world. Come on, guys. Done or what? Yes. Good, good. Good try, everyone. But the correct answer is the GM. It is very more is soya beans. Okay. Alright. So soya beans is herbicide tolerant soya beans. Okay. So it is going to occupy the largest area in the world. Correct. Okay, so production wise, okay, so you will think of this one in India. If you take, maybe BT cotton is little bit famous, right? Yes, but actually worldwide, no, soya beans only. And you know this Kellogg's fellow, no? Yes, this Kellogg's, it is also claimed to contain all this GM modified. So all the protein, it is coming from this corn as well as the soybeans. Okay. You thought maize. Okay, don't worry. Yes, biology. Come on. So clean gene technology. Name itself is good, no? Clean gene. Yes. Yes, Prachi. Absolutely. Clean gene technology. What do you mean by clean gene technology? Yes. It means it is going to create the transgenic plants with marker genes, or you remove the marker gene after the transformation. Name itself suggests. The plants are obtained with conventional breeding. Transgenic plants, it is obtained through plastid transformation, which is called as clean gene technology. Good try, everybody. I got mixed results. Okay. That's all. Good, everyone. Good try, everybody in the class. Which could be the correct answer, you know? Absolutely. It is going to be your option B. So it is clean, no. After you create the transgenic plant, you remove the marker gene. Yes. So you can just read it. Yes, because these topics are evergreen. It may come back anytime, and especially PBT, you have more and more repetitive questions. Okay, so this question is very, very important. Alright, so next class definitely I will plan with enzymes, numericals. Okay, so don't worry, I will definitely catch you up in another interesting session. Yes, absolutely, it means remove, that's all. Logically speaking. Alright, okay everybody, so I will catch you up in another interesting session. Take care till we meet. Bye bye all.